I'm going to introduce my graduate work, which is called APIR, a flexible and a powerful FDR control framework for aggregating peptide identification results from proteomics data. So as we all know, proteins are complex molecules that has 4 levels of structures. The primary level is basically the primaries consists of like amino acids, where you can see like each bean here is 1 amino acid residue. And the secondary protein structure refers to the local folding of polypeptide chains. So you can see there are 2 very common type of secondary structure, where one is the alpha helices, the other is beta pleated sheets. And the third type refers to the 3-dimensional folding of a peptide. So you can see locally this red peptide chain has alpha pleated sheets, but it could also fold in three-dimensional space, like the like here showing the picture. And many of the proteins are made up of only one single polypeptides. So these proteins have only these 3 structures. However, some of the protein could have more than 1 polypeptides. So these chains could like interact in the 3-dimensional space and form the quaternary protein structure. And in a typical shotgun proteomics database, in a typical shotgun proteomics analysis, we focus mostly on the primary structure. That is, we are interested in identifying the amino acid chains of proteins. And in addition, proteins also have post-translational modifications, which could change their functions. So we're also interested in those. So here is a workflow of a typical shotgun proteomics. So we first extract proteins from samples, and then these proteins will be digested and separated and measured by tandem mass spectrometry. And this mass spectrometry analysis will output mass spectra that encodes the chemical composition information of these peptides. And finally, we will do a data analysis using search algorithms to identify from these mass spectra what are the proteins in this sample, as well as the peptide sequences and the modifications. Okay, and to give you a more clear idea what database search algorithm does, so here I show a mass spectra of the glucose. So without going into further details, so the key message is that we could solve this mass spectra like we solve a mass equation. That is, this mass spectra contains the information that allows us to tell what chemical molecule this mass spectra comes from. Okay, and in a typical shotgun proteomics experiments, often tens of thousands of mass spectra are generated from different peptides. So it's impossible to manually solve each of them. Therefore, people have developed database search algorithms, which are basically computational tools to automate the interpretation of mass spectra. So these database search algorithms takes as input the mass spectrum from the MS analysis, as well as a protein database. So if your sample comes from human, then you would specify the protein database to be the human protein database. And these search algorithms, they output for each mass spectrum a peptide sequence from the database that matches the best. And the resulting match, we call it a peptide spectrum match, short as PSM. And also it will output in an internal matching score for this PSM. And this internal match score is dependent on which database search algorithm you use. So you could see different scores. I mean, you could see different ranking of this PSM in different database search algorithms. And also, these database search algorithms, they try to control FDRs, so they will also output a Q value for each PSM, as well as the proteins and the modifications. However, the best match does not necessarily mean it's a correct match. So errors could happen due to low quality spectra, imperfect data processing algorithms, or the use of incomplete protein databases. And it's estimated that in a typical run, 40% of the PSMs are false. And because peptide spectral matching is an indispensable intermediate for downstream analysis, therefore, it's very important to ensure the quality of this step. So we rely on a very popular statistical criteria, FDR, to help us ensure the quality of this step. So our goal is to do FDR control at this step. And here I show a sample output from a very popular search algorithm, Byonic. So you can see the first column that I highlight here is the identified or matched peptide sequence. And then the second column that I highlight here is simply the mass spectrum ID, as well as Q values here, which is used for FDR control. So to give you a little bit more idea how these Q values are computed, so the field relies on a very popular strategy, which is called the Target-decoy search strategy. And there are two implementations of this strategy. The most popular, the more popular one is the parallel search. So what it does is that it does two searches actually. So in the first search, they take the mass spectrum from the MS analysis as well as the target protein database. So the target protein database is essentially the original database. And then from this search, they will identify a set of target PSM. So you can see these mass so all these mass spectra will be matched to the target to the peptide sequences in the target database, as well as their internal matching scores. And in the second round, these mass spectra will be searched against a decoy protein database. So a decoy protein database is basically contains the artificially generated false protein sequences. So this search will provide a negative control that allows us to control the FDR. Okay. And the second implementation is the concatenated search, which is basically to combine a target protein database and a decoy protein database and do just one search. That is, we'll search the mass spectra against this concatenated databases, and we'll get the target PSMs as well as the decoy PSMs and their scores. So here is another output from Byonic that's from the decoy search. So again, here we have a column that's for the peptide sequences, but all these sequences are false. And again, we have a spectrum ID and we have Q values. So since the development of shotgun proteomics, many database search algorithms have been developed. And our collaborator, Dr. Leo Wang from City of Hope, he came to us that basically he wanted to combine these highlighted 5 search algorithms. But the thing is, the existing methods that can deal with these 5 search algorithms are not guaranteed to improve power. And moreover, some of the algorithms are not compatible. I mean, some of the search algorithms cannot be combined. So some of the methods that combine these search algorithms are not compatible with other search algorithms. So to help us, he generated so he wants us to develop a statistical framework that's universally adaptable to search algorithms with FDR control. And to help us validate our result, he first generates a benchmark dataset for us. So the first thing that we do is to investigate, so does it really make sense to combine database search algorithms? So we look at the total number of PSMs from these 5 search algorithms at FDR 1% and 5%. So we see that each individual search algorithms indeed identify unique PSMs. And in fact, Byonic alone can identify more than twice as many as the number of PSMs shared by all 5 search algorithms at FDR 1%. And at 5%, we can see MaxQuant identifies 4,000 unique ones, while the common PSMs by all these 5 methods is only 1,500. So the question is, what's the reason behind what we see here? So it could be that different search algorithms indeed capture unique PSMs, or it could be that these unique identified PSMs, they do not have very high quality, meaning that a lot of them may not be able to control FDR well. And finally, it could be a mixture of both these 2 effects. So to look further into this, we examined the 2 PSMs at these 2 target FDR levels. So our result here confirmed the unique advantages of individual search engines. And we also look at the FDR control. So we examined the target FDR from 1% to 10% and look at their empirical FDR. So this diagonal line means we have the empirical FDR equal to the target FDR. So as long as all points are below this diagonal line, this method controls FDR well. So we can see from the 5 search engines we have examined, Byonic, Mascal, Sequest, and MSGF, they all have a pretty good FDR control. However, MaxQuant, so the empirical FDR of MaxQuant exceeds this line by a large margin. So we see there is one particular search engine that's not reliable in terms of FDR control. So based on our benchmarking results, we developed APIR for aggregating multiple search engines with FDR control. And APIR has 2 functionality. The first one is the APIR select, which is simply an FDR control procedure for adjusting individual search algorithms. And it takes input the target PSMs, the decoy PSMs, as well as their Q values. So one thing to notice is that even though we take Q values as input, we do not necessarily use them for FDR control. And we just treat them as scores because sometimes you see some search algorithms may not output reliable Q values. And our APIR select is partly based on our recent work Clipper, p-value free FDR control on high throughput data from 2 conditions. And it's on bioRxiv. And the second functionality of APIR is APIR-merge, which aims to aggregate multiple search algorithms. So the key advantage of APIR is that it's universally adaptive to search algorithm with scores. And APIR-merge is guaranteed to identify more peptides than the most powerful individual search engine. And also, we can have FDR control with very mild assumptions. So I will use this diagram to explain why APIR-merge is flexible and powerful. So let's say we have this many PSMs and we have search engines 1, 2, 3 that we're interested in combining. And S1 here means spectrum number 1, P1 here means peptide number 1. And then you see, even though we have 11 spectrum here, some of them are matched to the same peptide sequence. For example, S2 is mapped to P2. S5 is also matched to P2. And then within each individual search engines, the darker color means it has smaller Q value, meaning that it's more likely a true PSM. And the gray color means that these matches are not output, or these PSMs are not output by this algorithm 1. Okay, and then APIR combines search algorithms sequentially. So what it does is that in the first round, it will apply APIR select or simply thresh Q values to each of the search engines here. And let's say these are what we found using these FDR control approaches. And APIR-merge will select the one that has the most number of unique peptides. So you see, even though from algorithm 1, we have identified five PSMs as well, but S2 and S5, they're matched to the same peptides. While algorithms from algorithm 3, we have identified distinct peptides. So we will pick this set as the first output. And once we do this step, we will exclude all the PSMs from search engine 3. That is, we will, excuse me. That is, we will just cross out these search engines just cross out the PSMs from search engine 3, and we will apply APIR select again. And let's say in this step, we have identified this many PSMs from search engine 1 and search engine 2. And again, we will pick search engine 1 because we have more unique peptides here. And finally, we will exclude PSMs from search engine 1. Sorry, here it should be 1. And then we will apply APIR select again, and we'll get this peptide. This match. And the final output of APIR-merge will be the union of these 3 sets. So you can see from this workflow that APIR-merge is guaranteed to output more peptides than the most powerful individual search engine. And also because these 3 sets, they're disjoint. And then if their FDP, if on each of them, the FDP is controlled under a value, then their union should also have the FDP controlled under the same value. So therefore, we will have FDR control with very mild assumptions. So I also mentioned that APIR select, it can be used to adjust individual search algorithms. So here I show the results of APIR select on the 5 search algorithms. So you see on Byonic, MassScout, Sequest, and MSGF, our method still can control FDR consistently. And on MaxQuant, we see even though APIR cannot exactly control the FDR under the target level, but we have already shown a big improvement in terms of the FDR control. And finally, I would like to show the results of APIR-merge on the benchmark dataset. So here, I coded these 5 search algorithms into 1, 2, 3, 4, 5, just to save some space. And because it takes sometimes hours or days to run each search engine, so we only show the combination of 3 search algorithms or 2 search algorithms, because that's what people will usually do. And then the first column here is that we look at the FDP of each combination. And then the second column is we look at the percentage increase in true PSMs compared to the most powerful individual search algorithms. And similarly, the increase in true peptides and the increase in true proteins. So here we compare APIR with scaffold. So there are several methods that could combine search algorithms, but scaffold is the only one that's compatible with all these five search algorithms. So you see, in terms of FDR control, APIR and scaffold does a pretty good job across all these combinations. However, in terms of power improvement, you can see scaffold is cannot guarantee to identify more than the individual search algorithms, and except for this case. While APIR, on the other hand, can consistently improve power. And we also see similar comparison results in terms of peptides and proteins. So one thing I would like to point out is that you see in this combination 1, 2, 3, 1, 2, or 1, 3, where we want to combine Byonic, Mascal, Sequest, you see a lot of times the power improvement is 0, and this is reasonable because it's widely acknowledged in the field that these 3 methods are very similar. But however, Byonic, the output of Byonic pretty much covers the output of Mascal and Sequest. So that's why when we combine these 3 methods, we see the power improvement is 0 because the output of APIR would just be the output of Byonic. So with that, I would like to thank my PhD advisor, Jessica, for her helpful guidance, and our collaborator, Dr. Liu Wang from City of Hope, and his previous research associate, Mei Lu, who is now a PhD student at USC, as well as his current research associate, Kyla.